back to Chapter 11, Lecture 2. We're going to talk in more detail now about Mendel's process and how he went about answering his questions, which involved how heritable traits were passed on from one generation to the next. So just to review, Mendel's process, he would start with two true breeding strains of pea plants, and he would remove the male reproductive organs from one plant, and then using a paintbrush pick up pollen from the other plant, cross-pollinate, and these two plants were his parental generation. The crosses would produce peas that he would then plant, and when those peas grew into plants, that would be the F1 generation, okay? And Mendel did this hundreds or thousands of times. So at the end of this PowerPoint, we'll see a chart that shows a bunch of the different traits that Mendel was looking for, but he was always looking, well, I say always, most of the time, at least in the beginning, he was looking at the inheritance of one trait at a time. And Mendel called the visible characteristics traits, and as a result of his experiments, he deduced that each trait was determined by two factors, and that each organism got one factor from each parent. Okay, sounds a lot like how we talk about chromosomes and genes and alleles. Okay, so these first two panels here are basically what we saw in the previous slide. So we start with the true breeding parental generation, cross-pollination to produce the F1. Well, because he was crossing two true breeding plants, and each parent donates one allele to the offspring, all of the F1 generation had the same heterozygous genotype because the only allele they could get from the purple flowered plant was a dominant P. The only allele they could get from the homozygous recessive white parent was a recessive P. So all of the F1 generation were heterozygotes. They all had one dominant allele, and therefore they all expressed the dominant phenotype. So Mendel's question at this point is, well, what happened to the white trait? Is the white trait gone forever, or will it show back up? So for the to create the F2 generation, he either allowed the F1 plants to self-pollinate, or he cross-pollinated only between other F1 plants, okay? So it was always heterozygote crossed with heterozygote, okay? The F2 generation is where things really start getting interesting, because in the F2 generation, that white trait showed back up, but it only showed back up a quarter of the time, okay? The rest of the time, the rest of the F2 offspring expressed the dominant phenotype. And so this F2 ratio, this phenotype ratio for the F2 generation is one of the most famous numbers in biology, and it is 3:1. That for every 3 dominant phenotypes that are expressed in the F2 generation, there will be 1 recessive phenotype. But again, this 3:1 ratio is only when heterozygotes are crossed in the F1 generation, okay? So F1 down here to F2, the genotypes would be big P, big P, big P, little p, big P, little p, and little p, okay? So 3 dominant phenotypes to every 1 recessive phenotype. Now, Mendel did this, like I said, with multiple traits. Your book has the full figure for the 7 different characteristics in pea plants that Mendel explored. I'm showing you just an abbreviated one with 4, okay? And so we talked a lot about flower color. We could also talk about seed color, seed shape, pod color. All of these traits have 2 different possibilities that are easily distinguished from one another. And all of them only show up in 2 possibilities, okay? So there is that dominant recessive relationship amongst these traits that Mendel was studying. Now, what I want to call your attention to are these numbers, okay? Mendel did a lot of counting, okay? So when we look at the traits, that we can describe in words, that's qualitative data. And in fact, Mendel wasn't the first person to do these experiments. All of these true breeding strains of plants existed. Other people had done these crosses, but they just were looking at the qualitative data, what they could see with their eyes. What Mendel did that was different is he counted. Can you imagine counting 6,022 yellow peas and 2,001 green peas for a total of 8,023 peas, individual peas, right? That's a lot of patience. But because Mendel was so rigorous and he had such attention to detail, and because he documented all of these counts, he was able to figure out these ratios. And notice they're not exactly mathematically 3-to-1, but they're really close. Okay, one of the things that new science students sometimes struggle with is, well, 2.82 isn't 3, so, these data would not support Mendel's hypothesis. Of course they would. Statistically, it's close enough. When you're dealing with living organisms, not every plant is going to survive. Not every plant is going to make its maximum number of offspring, okay? So when you're dealing with real living systems, you've got to have a little bit of grace if your numbers aren't perfect. That's just not real life, okay? Now, one of the other questions that comes up is, well, what if you come across a plant that has a dominant phenotype, but you didn't write down your label? So you're not sure who its parents were, or maybe you come across it in the wild and you're really curious and you want to know, is this plant that exhibits a dominant phenotype, is it homozygous dominant genotype or is it a heterozygous genotype? Well, the way to figure that out is through a test cross. So you would take your purple plant of unknown genotype and cross it with a recessive phenotype, so a plant with white flowers, because we know that if a pea plant has white flowers, it's homozygous recessive. Okay, so you're going to cross these two. Well, let's think about this for a minute. We know that when Mendel did his parental crosses of true breeding purple and true breeding white, all of the offspring were purple. We know that when he did his crosses of just the heterozygotes, that he got that famous 3-to-1 ratio. So when we do this test cross, if all of our offspring are purple, then we're going to know that our unknown genotype is homozygous dominant. If some of our offspring are purple and some of them are white, then we're going to know that our purple plant with unknown genotype is heterozygous. Well, you may be wondering, why isn't this ratio down here 3-to-1? Well, it's not 3-to-1 because both parents weren't heterozygotes. This second parent was homozygous recessive, okay? So that changes the allele frequencies in the offspring. And what we're going to talk about in the next lecture is how to set up these little boxes called Punnett squares. Now, chances are you've done these before, but since I am not looking at your faces and I don't know for sure who has learned what, in the next lecture, I'm going to go through Punnett squares as if you don't know how to do them. And there will be opportunities within the next lecture for you to pause